Hello, everybody. Welcome to week 10 of our course. This is physical and chemical control of microorganisms. And in this particular unit, we are going to be building on what you've already learned about pathogens, particularly bacteria, viruses, fungi this semester. And we're going to tie in things like cell wall properties, reproduction, DNA, RNA, life cycles, chemical characteristics of the cell wall. All these kinds of things are going to play into how are we going to control microorganisms so that they aren't overpopulating and harming us. So let's go ahead and proceed. All right, so really what we're thinking about here is reducing, it says controlling our degree of exposure to potentially harmful microorganisms. And this is a problem wherever we're at. So I'm working at my office desk, so thinking about my desktop. When I go use the bathroom, thinking about the microorganisms that are on door handles and the knobs on the faucets. Certainly whenever you're preparing food, taking something out of the refrigerator and feeling confident that you know that food is safe to eat. And it goes on to thinking about our drinking water, to thinking about, maybe lotions that you put on your face, frequently using that lotion and putting our hands on the spout of the lotion and then putting it on our face. Are we potentially then adding bacteria to that? So controlling microorganisms is essentially trying to reduce our degree of exposure through food, through products that we might use every single day, and those kinds of exposures from the environment. So whether that be the bathroom, maybe it's going to the doctor's office and there's people who are sick and coughing, and of course microorganisms on door handles and desktops. Okay, so we want to talk about first the methods of microbial control. And these are methods that we use outside of the body. So it's a very important distinction. It's an important distinction because when we're thinking about eliminating microorganisms, we don't want to harm tissues, right? So inanimate objects, whether that's maybe tattoo needles, or if you're getting your ears pierced, or if you have to have an endotracheal tube or something like that, IV, we want to know that those objects are sterile. They are completely free of any microorganisms. And you might remember that the cell structure of bacteria that's the hardest to eliminate is the endospore. So completely free of microorganisms, including the endospore. Disinfection is essentially any process that is going to destroy vegetative pathogens. So we're thinking about fungi, bacteria, viruses, but not endospores. So we're disinfecting, we're reducing the amount of microorganisms on a surface, but disinfection techniques don't eliminate endospores. We have to use more specific techniques for that. Okay, decontamination. So this is also for inanimate objects. So essentially sanitization. So we're thinking about cleaning surfaces. We're thinking about anywhere where they're preparing food or packaging food. So whether that's beer or whether that's milk or whether that's whatever, that essentially those surfaces are clean and free of, let's see, so I'm just looking at my notes here. So it has a clean surface. We're going to mechanically eliminate or wash surfaces with soap or detergents. So we are essentially ensuring that we don't have a source of contamination on those materials, whatever it is, food, for example, that we're packaging. Okay, antisepsis. So antisepsis is reducing the number of vegetative pathogens from living tissues. Now, it's really difficult to make living tissue sterile because of the nature that it's alive. So our cells are living and they're reproducing or dividing and degrading and all these things. So the best that we can do usually on human surfaces is what we call antisepsis. So anytime that you've had an IV put in, they're going to, clean your arm with alcohol. If you go to give blood at the Red Cross, they're going to use hexadine, chlorhexidine. So they're going to clean the surface of your arm really well. So we can get pretty close to a sterile environment, but it's not considered sterile because it's on living tissue. Okay. So moving forward, I don't want you to memorize this table, but essentially this does a really nice job comparing these different kinds of techniques. So we are going to spend some time going through various methods of control, both physical and chemical, and I believe you'll have a better idea of how this works as we move along. But again, you can see the definitions here. Sterilization, the process that destroys or removes all viable microorganisms, including viruses, but also endospores. So some examples there are heating or using an autoclave. So I will go through that a little bit. And then sterilants. So these are a chemical control capable of destroying endospores. Again, disinfection, a physical process or a chemical agent to destroy vegetative pathogens, but not necessarily the endospores. We can use bleach, which we've certainly done this in household situations or cleaning situations, restaurants, hospital bathrooms, and so on, but we also have iodine and heat. Decontamination, which is also the same as sanitization, is essentially, as I mentioned before, a cleansing technique that's going to mechanically remove microorganisms and other debris to a safe level. So we may not get it to be 100% sterile, but we're definitely keeping surfaces, utensils, bottles, if you're bottling beer, all the equipment used to do that should be decontaminated or sanitized. So we can use soaps, detergents, and of course, dishwashers are pretty good at that. And then the last one, antisepsis. So we just talked about that, using alcohol on your deltoid before giving an intramuscular injection, for example. Chlorhexidine on your arm. So when you get blood with the Red Cross, we can ensure that that blood is going to be free of pathogens and, well, of course, microbes. So you know that we can only decontaminate living tissues because they are living. So a little bit of review from the previous slide. I quite like this particular table because what you're going to see is we are going to talk about first chemical agents of control, and we will have at least one mechanical method of control. And then here we have the chemical agents. And you're going to see that whether it's physical, chemical, or mechanical, that we can have sterilization techniques that fall into each of those categories. We see disinfection is going to fall into at least 2 of those categories, antisepsis, only one of those categories, and then decontamination, only in one category. So it's going to help you if you organize your thoughts around this, so it doesn't seem like just so much material being thrown at you. To help you with that, I have put together some tables that is in the Week 10 lecture handout. And so if I were you, I would spend some time pausing the video and then filling out the table that's related to that so that you can consolidate a lot of wordy material into some simple words in a table that essentially allows you to compare and contrast methods. Now, what you will see is that I'm going to be doing the physical agents and the mechanical agent, and then we will have a guest speaker, Professor Dr. Brenda Gustin, who has been the owner of this course for 28 years, I believe. And so she's going to be doing the lectures on the chemical controls. So I will do the first few, and then you'll get to see her. And you've probably seen her here and there throughout our lab work as well. Okay. All right. So relative resistance of microbial forms. So the story is that depending on the properties of the microorganism is going to depend on how easy it is to eliminate them or reduce them. We know that endospores are our biggest challenge, right? So that is going to allow us to set the standard for essentially the most stringent methods is if it can eliminate the endospores, all right? So that's essentially what this is telling us. And when we think about sterilization, we know that we're also destroying the endospores, which again, in a living organism, we can't sterilize and wouldn't recommend sterilizing the large intestine, for example, of someone who has Clostridium difficile. Why would you want to do that? In fact, probably what allowed C. diff to colonize is being on antibiotics, like those big gun antibiotics, for such a long time that it has eliminated the beneficial gut bacteria. When we do that, we're essentially allowing it to be colonized by anything that gets there. And C. diff is really good about getting there. Okay, so I like this figure better than the previous slide because the previous slide had lots of words, but let's take a look. So if we essentially take those microorganisms that we're most worried about controlling, and we put them on this spectrum essentially of the least resistant to our controls, okay, to the most resistant, then we would see that our enveloped viruses are the easiest thing to treat. So we see SARS-CoV or COVID was essentially in this category. Then we can see most of our gram-positive bacteria are next. There's properties, of course, of gram-positive bacteria, that lipopolysaccharide layer that is going to allow them to take up chemicals and be essentially, well, it's just a chemical or physical. They're just easily killed, all right? So then we go on to our non-enveloped viruses, fungi and fungal spores, gram-negative bacteria. We've got some protozoans. The cyst form, clearly more resistant than the living active form, the trophozoites. And so it goes down. So we see Mycobacterium here. So this is the causative agent, or at least the genus Mycobacterium tuberculin. So these bacteria have a waxy wall, so it makes them very, very resistant to chemical controls, to chemical and physical controls. Then we have our endospores. Now we know that prions are relatively new to science, and so to control them, we're starting to understand that. So we put prions up there, and when I'm talking about microbial control agents, the microbes I'm talking about are not including infectious protein particles. So prions are kind of out of the normal way of thinking about microbial control agents. Okay, this slide is giving us an idea of the comparative resistance of bacterial endospores and vegetative cells to our control agents. So essentially, if you take a look at the methods on the left-hand side, so here's heat, which is a physical control, radiation, which is a physical control, sterilization gas, chemical, and sporicidal liquid chemical. So if you take a look at the vegetative form, required to destroy vegetative forms in that third column over, you're going to see, okay, to destroy the vegetative forms with heat, we need essentially 80 degrees Celsius, and we're thinking about 20 minutes, maybe 30 minutes. Radiation, you got a dosage there, what they call 1,000 grays, of course, sterilizing gas, and the sporicidal liquid. So you go on down, and so to kill endospores, so this is like a relative resistance, right? To kill endospores, we're going to have to heat those endospores at 120 degrees Celsius. We can only do that with special machines, and the one that we frequently use or commonly use is called an autoclave. So in this case, our endospores are 1.5 times more resistant than the vegetative cells. So here we are having to heat at a higher temperature, not necessarily longer, but a higher temperature to kill those bacterial endospores. Radiation, you're going to see that we're going to have to increase that dosage by 4 times to kill the endospores compared to vegetative forms. Sterilizing gas, we've got to essentially increase that from 700 milligrams per liter of ethylene oxide to 1,200 milligrams per liter. So essentially the endospores are 1.7 times more resistant to this control compared to vegetative forms. And then lastly, you see the sporicidal liquid. Okay, so we need to differentiate in the last 30 seconds, agents versus processes. So sterilization and disinfection are processes. And then essentially we have a variety of agents that we're going to use in those processes. So remember, sterilization, the complete removal of microorganisms, including endospores, and disinfection, radically reducing on living surfaces.